in a excitatory postsynaptic potential versus an inhibitory postsynaptic potential. And I was talking about the neurotransmitters that are usually associated with these two types of greater potentials. So glutamate is usually a neurotransmitter of an excitatory postsynaptic potential, whereas GABA is the inhibitory neurotransmitter. And so we talked about an excitatory postsynaptic potential being a positive change to the membrane potential, being a depolarizing event, and making it more likely, more favorable for an action potential to happen. And we want to be thinking about the reference point of the resting membrane potential here. So remember, we're trying to get from -70 to -55, and depolarizing events do that, right? They get us a positive change and get us closer to that threshold value. Inhibitory postsynaptic potentials are going to be less likely to produce an action potential. They're going to do one of two things. They can hyperpolarize the membrane, which is a negative change in the membrane potential. Think about a change that makes the membrane potential more negative, but they can also resist a membrane potential change. Again, making it harder for an action potential to happen. So we can hyperpolarize the membrane, which is a negative change in itself, or we can resist the membrane potential change by making it hard to depolarize the membrane and resisting any changes that would get us closer to threshold. And I'll kind of explain what that means with chloride as an example. So here we're again comparing the fast versus slow response, and I'm going to go over this just to kind of summarize what we talked about on Wednesday and just to clarify any misunderstandings. But we've got the fast response. This is where we have an ionotropic receptor as the channel, and because the receptor is also a channel, this is a very immediate, very quick, fast response. So the neurotransmitter, if you remember our series of events after the action potential arrived at the axon terminal, the neurotransmitter was released via exocytosis. It crossed the synaptic junction. It's going to bind onto this receptor, which is also acting as a channel. And so the binding of this receptor to the channel opens up the channel. Remember, this is a ligand-gated channel. So binding of that neurotransmitter will open it up, and that is going to allow the flow of ions. Now, depending on what channel this is, that will determine if it's going to be an IPSP or an EPSP. So if it's a sodium or calcium channel and we open it up, that will be an EPSP. But if it's a sodium or calcium channel and we close that channel, that's going to be an IPSP. So we want to be thinking about the events that are brought about if we open the channel, and then also thinking about what could happen if we close that channel. In terms of a slow response, the movement of the ion and the generation of an IPSP or EPSP is the same, but the steps to get there are much longer. It takes more time. And so this is why we refer to this as a slow response. There are a couple of intermediate players involved. It's not just boom, opening the channel and done. We've got the binding of this receptor, the binding of this neurotransmitter to a metabotropic receptor, activating this G protein that's coupled to the receptor such that the alpha subunit dissociates and floats downstream in the membrane. It's going to activate another enzyme, and that enzyme will produce some second messenger, usually cyclic AMP. Cyclic AMP will enter the cell and it will help to bring about that cellular response, either directly by activating some other enzyme, or by activating that channel directly, which will open up or close and bring about an IPSP or EPSP respectively. So as you can see, there are a lot of steps involved in the process, and this is why we refer to this as a second messenger or a signal transduction mechanism, because this signal has to be passed from one player to the next in order for that channel to be opened or closed and for that IPSP or EPSP to be generated. And so that's the essential difference between the fast versus that slow response. And so when we look at the inhibitory synapse, here we said that we can either open up a chloride channel or a potassium channel. And because of the movement of those ions, we would generate an IPSP, specifically because potassium is moving out of the cell, positive charge is leaving the cell. That would decrease the membrane potential or hyperpolarize the membrane potential. And then chloride as another example, because if we open up a chloride channel, specifically on a neuron that has active pumping of chloride. So if it's a neuron that has active transport of chloride out of the cell, when we open up that chloride channel, chloride will want to diffuse into the cell according to its gradient, and that will be negative charge entering the cell, which will also be a hyperpolarizing event, right? That will decrease the membrane potential. Now, chloride can also stabilize the membrane potential. Okay, so it can be an IPSP, but it can also resist any membrane potential change. And that will be on a cell that doesn't have active pumping of our chloride out of the cell. So here on this neuron, there's no active transport of chloride out of the cell. And so chloride is pretty much just moving at equilibrium in the cell, okay? And so at that type of, on that type of cell, if we open up a chloride channel, it's pretty much resisting any positive change to the membrane potential. And we can see that our voltmeter stays pretty flat, even though we open up this sodium channel, which is shown in red here. So let's say we have one synapse that opens up a sodium channel, sodium is entering the cell. For every sodium that enters the cell, one chloride enters the cell. And it pretty much cancels out that membrane potential change. The positive charge is canceled out by the negative charge of chloride. And so in this way, on a cell that doesn't have active transport of chloride, we resist the membrane potential change and we prevent depolarization. In other words, we make it really hard to fire an action potential in this cell. Okay, and sometimes that's needed. Sometimes it's important to resist any membrane potential change to make it harder to fire an action potential, and that's what chloride can do on this type of cell. Okay, so let's look at IPSPs here again as an example, just to remind ourselves of the flow of events. So let's say we are firing action potentials on a neuron. We increase the frequency of action potentials. And we spoke about this idea where action potentials do not change. We cannot change the magnitude or the duration. And so the way that we can change different signals using action potentials is by changing the frequency, how fast those action potentials are generated. And so a higher frequency of action potentials translates to more neurotransmitter being released. When that neurotransmitter is released, it's going to cross the junction, bind to the respective receptors, which may open or close. So again, here we want to be thinking about the action, but what would happen if we close that channel as well? So an IPSP could be brought about by opening up what type of channel? What type of channel did we say we would open in order to generate an IPSP? Excellent, yes, so potassium or a chloride channel, perfect. What type of channel would we close to generate an IPSP? Yes, that's correct. So calcium or sodium, right? So again, here we want to be thinking about what channel would open to give us this type of greater potential, but also the reverse. What type of channel would close that would bring about that same membrane potential change? And so opening or closing respective channels would either increase or decrease respective ion permeabilities, and that would lead to a greater or lesser ion flux. So for example, greater flux of calcium, of chloride or potassium, lesser flux of calcium or sodium, and that would lead to our hyperpolarization. So this is how a higher frequency of action potentials leads to a greater degree of hyperpolarization, because increasing the frequency increases the amount of neurotransmitter that's released, which pretty much increases the degree of the signal that's translated onto that subsequent neuron. Okay, and that is how we can sort of grade the signal that is transmitted using action potentials, although action potentials are not graded themselves. We can modulate or manipulate the frequency of those action potentials. All right, so let's look at these checkpoint questions here. We kind of talked about this on Friday, so these should be pretty straightforward. Let's look at question 1. So activation of which type of receptor, ionotropic or metabotropic, produces the faster response? So which of these two types of receptors is associated with a faster response? Excellent, yes. So the ionotropic receptor is going to be the faster response. And just to go a step further here, this is because the channel is the receptor. The channel is also the receptor. And so binding of that ligand, that neurotransmitter, to the receptor will immediately open up that molecule because it's the same exact structure. Excellent. Question 2, what is the role of G proteins in the operation of metabotropic receptors? So what is the role of a G protein in the metabotropic mechanism? Okay, yeah, so it's going to activate the enzyme. That's absolutely correct. So we said a part of that subunit would dissociate off and it would activate the amplifier enzyme that's responsible for generating the second messenger. So it's pretty much one of the intermediate players that will pass that signal from the receptor once the neurotransmitter binds to the enzyme that will help to generate the second messenger. In question 3, if a G protein activates a second messenger that closes sodium channels, will this activation produce an EPSP or IPSP, and will the response be fast or slow? So kind of a two-part question here. Will this be an EPSP or IPSP, and will it be fast or will it be slow? Excellent. So this will be a fast response. Excuse me, this will be a slow response, and this will be producing an IPSP. Because we're closing sodium channels, we prevent the flux of sodium into the cell, we make it harder to fire an action potential, harder to depolarize that membrane potential, and so that will be an IPSP. And because we mentioned second messenger and G protein, these two players are only seen with the metabotropic receptor, which is the slow response, okay? So IPSP and slow response. Okay, so I have a question here. Are inhibitory responses always associated with slow responses? And that's a no, okay? Right, we can have a fast response be an inhibitory response or an inhibitory postsynaptic potential if the channel that's opened up is a chloride or potassium channel. And if the channel, so we can have an ionotropic receptor that is also a chloride or potassium channel, and that would be a fast inhibitory response, if that makes sense. So the ionotropic receptors, we said they're also acting as channels, and depending on what channel they are, what ion flux they're allowing, that is going to determine the type of graded potential. So having a fast response does not necessarily mean it's always an IPSP or EPSP. The type of channel that's opened or closed and whether or not it's opening or closing is what will determine the IPSP versus EPSP. Okay, I hope that's clear. Any